Again, welcome and thank you for joining. I guess good morning, good afternoon, depending on where you are on the globe. We have just over 100 people on the call just now, so we will make a start. And I would like to wish you a very warm welcome to this webinar, as I said, organized by the BioPhorum community. I am your host, Alice Legat. I am a facilitator at BioPhorum. And today we are talking about environmental monitoring performance qualification in new facilities. Well, as many of you probably know already, this webinar has been prepared following the publication of the BioPhorum paper titled Environmental Monitoring Performance Qualification in New Facilities, an Industry Harmonized Approach. And it is the outcome of a great collaboration between all the authors that are listed here. They are members of our environmental monitoring discussion group at BioPhorum. And I would like to thank them very much for their exceptional commitment and work with this paper. Just a reminder that the paper represents a consensus view from the individual authors and should not be attributed to their employer. Also, the paper is available to download for free on the BioPhorum website, and you can use this QR code to download it as well if you want. Okay, so because we prepared this webinar based on the paper, hopefully by the end of this session, you will have a good understanding on the industry-led approach that we are proposing for EMPQ in new facilities. You will learn more about key elements of an EMPQ, all the way from prerequisites up to post-qualification activities. You will also hear 2 case studies, including recommendations, lessons learned, and best practices from 2 of our members who are renowned global biopharma manufacturers. And as I said, during the Q&A, you will be able to engage with the experts who have written the guidance, have your questions answered. And more generally, I would say that this webinar is a great opportunity to share knowledge to ensure consistency and robustness in environmental monitoring practices. So I am delighted to introduce our amazing speakers for today's session. Gurpreet Ganda is Director of Quality Control Microbiology at Roche. Manshi Patel is Associate Director of Global Quality Microbial Quality Sterility Assurance at Merck & Co. MSD. And Luyen Nguyen is Global Sterility Assurance and Microbiology Lead at Takeda. These 3 speakers will be supported during the Q&A by 2 panelists, Dawn Watson, Executive Director of Global Microquality and Sterility Assurance at Merck & Co. MSD, and Hilary Chan, Global Sterility Assurance and Microbiology Lead at Takeda. So thank you very much to the 5 of you to be the presenters and panelists today. In terms of the structure of the presentation, I will start very briefly by giving an overview of the BioPhorum collaboration. Then we will go ahead with the presentation of our guidance. And this will follow then 2 case studies that I mentioned earlier. 1 case study delivered by Merck & Co. MSD on drug substance facility, and 1 case study delivered by Takeda on cell therapy manufacturing facility. And finally, we will have the Q&A parts. And I remind everyone again, if you have any questions during the presentation, please use the Q&A box on the top of your screen. Well, so briefly, before I hand over to our first speaker, what is BioPhorum and what are the objectives of the BioPhorum collaboration? BioPhorum creates an environment and provides a platform for the biopharmaceutical companies to collaborate, share knowledge, and combine expertise to solve common challenges and problems and find solutions together for the benefit of the industry. So the BioPhorum community is organized in different forums that focus on different areas of the industry so that each forum would align with the interest of our members. And this webinar enters into our fill finish forum, whose mission is to develop and implement best practice processes in sterile filling operations. And so in fill finish, we have several work streams tackling specific topics. And one of these work streams is the environmental monitoring discussion group or team. And the EM team has been collaborating since 2016. They focus on defining, sharing best practices, guidance, and tools for EM in modern facilities. And at the moment, we have approximately 50 SMEs from more than 20 leading companies in the industry. Now, without waiting any further, I would like to hand over to our first speaker of today, Luyen Nguyen, Global Sterility Assurance and Microbiology Lead at Takeda. Luyen will provide an overview of the regulatory requirements and standards related to environmental monitoring. So over to you, Luyen, and thank you very much for being with us today.
Thanks so much. Thanks for the introduction and providing the overview of the presentation. So before discussing the harmonized guidance to EMPQ for new facility, it is important for us to understand what are regulatory requirements and expectations with regards to environmental monitoring. This is a limited list of requirements and standards from different regulatory bodies. I won't go over each one of them, but wanted to share. FDA guidance for industry stipulates a well-defined written program evaluating the quality of air and surfaces within a clean room environment. EU Annex 1s require a monitoring program that effectively evaluates the state of environmental conditions of clean room, including airlocks and pass-through. Thus, it is critical for us to have a robust EM program. And an important step in developing a robust EM program is performing an EMPQ. Additionally, there are industry harmonized guidance to provide further insight on how this specific regulatory requirement can be met. This includes PDA Technical Report 13, which discusses about fundamentals of environmental monitoring program, including performing EMPQ. Prior to performing EMPQ, the monitoring site must be defined, and BioPhorum published a risk-based tool that can be used in determining the monitoring sampling location. And for the topic of today's presentation, BioPhorum published a harmonized guidance to EMPQ for new facility, which provides detailed guidance to how to perform EMPQ from prerequisite to sampling requirements and post-qualification activity. But first, let's start with some basic concept. What is EMPQ and its purpose? When do we perform it and why it's important? So what is EMPQ and the purpose of EMPQ? EMPQ is a multi-phase process that evaluates the performance of clean room across different occupancy states based on a predefined parameters. It confirms the effectiveness in combination of cleaning and disinfection program, personnel gowning, and material transfer process. It also evaluates the aseptic behaviors of personnel. When is an EMPQ needed to be performed? It is performed as an initial qualification of the facility. It is also performed post-major modification to facility that impacts their flow, personnel, material movement within a clean room. EMPQ may be required as a result of an adverse trend that lead to changes to facility, disinfection, and or gowning program. But for today's presentation, we will focus on the initial qualification. Why is EMPQ important? I kind of alluded to this earlier. EMPQ is an important step in establishing and qualifying a robust EM program, which is a regulatory requirement. A robust EM program ensures the facility is in a state of particulate and microbial control. I now will hand it over to my colleague Gerbert to tell you how to perform an EMPQ.
Thank you so much, Luyen. So the next up is the EMPQ and the how portion of it. I do want to mention before I delve into the how part that the guidance documents that my colleague Luyen just mentioned, they describe the what, when, why, but the how part is a little bit light on details. So this effort or this guidance is actually really incorporating all the good practices from the companies that were participating and really coming up with the how in a little bit more clearer sense. And in the how section, I'm going to be talking about the prerequisites for the EMPQ. How do you establish alert levels and action limits? What are the sampling requirements? What are the acceptance criteria? And then we'll finally talk about the final report and area release. So the first up, prerequisites for the EMPQ. So prior to execution of EMPQ, there are various items that must be finalized or defined. You can see on the list here, like there are some clean room qualification maximum occupancy that are being discussed here. So it's important that before we dive into the EMPQ that you have your IQ and OQ done. There is a list of non-exhaustive items that needs to be done for that. I'll not go over those, but those are the items that needs to be done before the EMPQ is performed. It's also important that the maximum occupancy is defined. Maximum occupancy is how many number of people can be performing the activities in the manufacturing areas without impacting the state of the control. And EMPQ is a great way to actually check that as well. So, but you need to define that how many people would be needed in every room before the EMPQ starts. It's also important the clean room classification is done or completed before the EMPQ starts per ISO 14644. And any equipment that is inside the facility is installed, qualified, and calibrated. So this list continues in terms of other aspects like contamination control strategy should be defined. Any risk assessments that you might need should also be defined or completed before you delve into EMPQ. Your SOPs for operation, for the transfer and flow of equipment, material and waste, housekeeping, gowning, personnel flow should all be in place and approved. It is also important that your EM sampling methods, what kind of media you're going to be using, what is your growth promotion strategy, what is your incubation strategy. These are also defined before the EMPQ starts. In addition to that, any operators, people who are going to be performing this EMPQ, they need to be trained on the procedures, aseptic behavior, and any controlled contamination training. Your disinfectant efficacy studies should also be there, which kind of will help you understand what disinfectants you're going to be using. And this study will basically confirm that those disinfectants are indeed effective. You should also have your final approved and facility maps, approved facility maps in place before the EMPQ starts. So last but not the least, it's also important that the sampling sites that you're going to be testing during the EMPQ are defined. I'm not going to go in a lot of detail because there is a paper and a webinar that's available from BPOG about the EM risk-based assessment, which goes in a lot of details of how to perform it, how to go do gridding and looking at the risk factors, and then determining the sampling locations. But the bottom line for the EMPQ is your sampling locations should be defined before the EMPQ starts. So before again diving into the sampling requirements, it's important that you have your alert levels and action limits established as well. Now, what are alert levels? Alert levels are your early warning of any potential drift from normal operating conditions. They can provide they can really provide like early detection for any potential issue at a sampling location, in a room, in an area. So you need to have an alert levels defined. Now, one of the challenge with the new facility is then what to put as an alert level. So the recommendation is if you have a comparable qualified facility that exists at your site, you can take alert levels from that existing site prior to having any data. Or if you don't have any comparable facility, then you can set the alert level at 50 to 60% of your action limit. But once the PQ is completed, the alert levels should be reassessed at that point. So action limits are relatively easy to have just because, the regulatory requirements, they do have, action limits that you should be applying with respect to the classification. So the recommendation is to look at EU Annex 1, FDA guidance to come up with those action limits exclusion. It is highly recommended that your SOPs should have these in there, which define what your alert levels are, what your action limits are, and at what frequency are you re-evaluating your alert limits. So once all of this is done, the prerequisites are completed, you have your alert and action limits. It's important to understand that EMPQ is done in two occupancy state. One is at rest and one is in operation. So before diving into what are the requirements, I just want to spend a few minutes on defining or sharing with you what at rest and in operation means. So at rest is like any condition where your clean room classification and everything is complete, your equipment is there, but there are no personnel present. You're really checking the efficacy of technical and procedural requirements or your engineering controls. For example, your HVAC is working, your cleaning and disinfection program is in place, but there are no people and no process that's happening in there. It is recommended to perform at least one sampling set of at rest occupancy state. In contrast to that, in operation is everything that I just mentioned for at rest, plus your people and processes are happening as well. And what you're really looking in operation is that what is the potential impact of having people and processes on the status of your clean room? It is recommended to at least have minimum of 3 sampling sets. I will go over in a little bit of a detail in the subsequent slide of why 1 and 3 were recommended for the sampling sets. But in short, it's really accounting for any variability that you might have over in the production activities. And as you can know, like people are the biggest contributor to your clean room microbial contamination. So you're really seeing like what kind of variability are they bringing in. So in operation sampling is helping you with that. It's allowing you to perform EM sampling and getting a representative shot of when people and processes are ongoing in your clean room. So with all of that done, with your sampling locations in place, hopefully by now you have done your risk assessment and you know what sampling locations you're going to be looking at, what is your occupancy state, sampling types, number of sets. Here's a little pictorial definition of how then you should proceed. So at first, after the clean room is released from IQ and OQ and the cleaning is done, the first set of at-rest monitoring can be performed when there are no operators and no activities that are happening. Once that's done, another set of cleaning and disinfection should be done. And now you need to introduce your routine operations. Your maximum occupancy of the room needs to be there. So have like maximum number of people who you're going to be needing for your operations. If your company has 3 shifts, for example, then do consider shifts, like having these 3 sets across 3 different shifts. So have it in that shift, and while people are performing operations, while those rooms are maximally occupied, perform your first set of in operation. And then this will continue for 3 times. The BPOG does recommend minimum of 3 sampling sets for dynamic monitoring or in operation monitoring. Once these sampling are done, you have the results. The clean room can be then released for the routine production. This slide is just taking you through what I just described in a slightly different form. So again, this is just reiterating that you're performing 1 sampling set at rest, 3 sampling sets in operation, but you're doing it for non-viable, viable surfaces, active air, and passive monitoring air. So here's a slide that will talk a bit about what is the rationale of picking 1 sampling set for at rest and 3 for in operation. So when we're doing at rest conditions, since there are no people, which are the biggest contributors of any contamination that could happen in the clean room, um, the risk is very low of ingress of organisms, especially once you have done cleaning and disinfection. So this is really informing you about what is the overall levels of particles. And then in general, there's a very low risk of any potential impact to the state of control when it's only equipment and the cleaning and disinfection has been done. Considering that low risk, only one set of sampling is recommended for at rest. For in operation, on the other hand, 3 sampling sets is recommended. The reason for that is like you're really looking at the variability when it's in operation, because now you've introduced people, you've introduced processes. So now you're looking at the variability. You're really looking to demonstrate the reproducibility and really making sure that your clean room can like demonstrate the consistent and meaningful results with enough reproducibility there. So, and in addition to that, this is also regulatory requirement, and this is 3 number is a magical number because of the statistical significance that it provides, as opposed to 1 or 2 sets. So once you have performed your EMPQ, now there are this slide goes over the acceptance decision. This is just an example, and there are broadly speaking, there are 2 things that can happen. One is that you collected the samples, they were all valid, they met the action limit, or they were below the action limit, sorry, and then they were below the alert limit. In that case, you have completed your PQ, you can document and proceed to perform the final release. But this table also tells you that in case you don't get a valid results, then what to do. In this case, the recollection of the samples is recommended. If you have action limit, then what should you do? You're expected to investigate, have CAPAs in place, and then document that event in your final report. If you exceed alert limit, you need to be performing trending, seeing if there's an adverse trend or not, what additional action needs to be taken. In either case, like once all of these actions are done, the sample evaluation is completed, and then you proceed on to your final report. So the last section of this is final report and area release, and I'll hand over to my colleague Manshi to take you through this section and the case study. Thank you.
Thanks, Gurpreet. So after the results are reviewed and assessed, there should be a final report that includes a summary of the results, any deviations to the protocol, along with the impact to the EMPQ, investigations for the alert level, action limit excursions, as well as missed or invalid samples, any associated CAPAs, and confirmation that the maximum occupancy and the representative activities were performed during the in-operation sampling. And finally, an overall conclusion regarding the EMPQ must be included in the final report, confirming that all samples were collected and are in compliance with the acceptance criteria. Additionally, prior to the release of the manufacturing area for GMP use, you want to make sure that all associated testing are complete and any impact to deviations must be assessed, as well as quality or the area owner minimally should approve the final report. If the report cannot be completed prior to manufacturing, an interim sampling strategy should be employed utilizing the EMPQ sampling points. An interim report should be generated with the indication of whether the EMPQ acceptance criteria were met, and the final batch release must be contingent on approval of the final EMPQ report. Once the qualification activities are complete, routine program should be established based on the environmental monitoring, risk-based assessment, and the results of the EMPQ data. Additionally, EMPQ, a strategy for requalification, should be established for periodic evaluation to demonstrate compliance, and this should be based on risk depending on the level of change control, whether there were any planned, unplanned events, adverse trends, extended shutdowns, or any breach of integrity to the facility. Now we move on to the case studies, 2 case studies that were mentioned earlier as part of this, where the EMPQ guidance from the paper was applied. The first one was applied to a drug substance facility that comprised of grade C and D rooms. So this is a low bioburden process that serves as a sterile final production step, producing a sterile bulk substance. And the process of execution was largely aligned with a harmonized approach stated in the paper. Prior to the EMPQ execution, we noticed that a couple of deficiencies on the floor were observed, such as sanitization execution was not executed correctly, as well as found that the roller door on the non-classified side was damaged. As part of this recovery, we made sure to incorporate these considerations as an insights gained before the EMPQ. We made sure that all personnel involved in sanitization are adequately trained for the task to ensure that they possess the necessary level of knowledge and skills to perform sanitization, as well as it is equally important to verify that the equipment that is used for sanitization is in good condition and suitable for the intended purpose, because deficiency of these processes could lead to incorrect application of disinfectant and ultimately impact the EMPQ. Another important factor is to perform Gemba walks to ensure that all checklists are completed to help identify and address any potential issues that may compromise the integrity of the area. Some of the examples are issues with the sealing of the panels, verifying the proper functioning of door automation, and any non-essential tools to be removed. As again, any deficiency of the above could lead to cross-contamination and impact to the EMPQ. In order to ensure integrity of the EMPQ, it is important that EMPQ be performed as a solo activity. This means that no other conflicting activities should take place concurrently that could introduce contamination. That is, folks should not be performing any process simulations as well as performing any commercial production concurrently. There should be limit access to the impacted area to only essential personnel including those involved in the execution. Consideration must also be given to the duration and the maximum number of personnel present, as stated earlier, performing or simulating routine operations at work. And additionally, effective communication between the department is important in order to ensure that all departments associated with the impacted area are aware of the dates, the execution plan, and the limitations on personnel access. Failure to address this could result in cross-contamination, contamination ingress, as well as compromise the results and have the potential to impact EMPQ. As stated earlier in the presentation, as part of the execution of the EMPQ, all prerequisites, IQ, OQ, clean room classification, risk assessments were completed prior to the execution, as well as alert levels were established based on the historical data from a comparable manufacturing facility on site that had similar processing steps, similar classification, similar cleaning and disinfection, and personal and material controls. And the action levels were set per regulatory requirements. So this is a schematic of the facility. The sampling locations were selected based on the environmental monitoring risk-based assessment, utilizing the BPOG harmonized approach stated earlier. In particular, we used settle plate locations in grade C and D that were based on the consideration for higher levels of activity, including the material and personal flow. This is in a detailed overview of the execution of the sequence of steps that were carried out to complete the EM sampling, starting with one set of at-rest sampling set, followed by 3 sampling sets of in operation. And note that we were able to utilize the EMPQ data to further support the routine cleaning and disinfection program. At the completion of the EMPQ, all results were reviewed to determine which sites will be included in the routine monitoring program, and the EM risk-based assessment was used to determine the routine monitoring sites that were deemed high risk, optional for areas that were assessed as medium, and only taken at low area when there were significant recoveries for the EMPQ. And the conclusion was that all static and in operations testing results were within specification and EMPQ was executed smoothly with no concerns. And now I'm going to hand it over to my colleague, Lewin, who will go over the case study from Takeda.
Thanks so much. So, yep, our next case study is EMPQ performed in a cell therapy manufacturing facility. This facility is used to manufacture both allogeneic and autologous cell therapy product. It is an open aseptic process that is conducted in a grade A biosafety cabinet with a grade B background. It is of note that this EMPQ was performed during the development of the BioPhorum EMPQ document, but the approach aligned with the harmonized guidance. Prior to execution of EMPQ, a list of prerequisites were completed and signed off. So HVAC was commissioned and clean room operation qualification was completed, including classification. Maximum occupancy was defined, routine cleaning and disinfection program was implemented, and EMRA was completed. To establish alert level, we set it to 50% of the action limit. The action limit is aligned with EU Annex 1, but EU Annex 1 does not define the action limit for grade D in operations, so that was established internally. Shown here is the EMPQ plan. We've performed 3 sets of at-rest monitoring, including all different methods of sampling, total airborne particulate, microbial surface, and active as well as passive air monitoring. And 3 sets of at-rest monitoring was above and beyond the minimum requirement of 1 set per the harmonized guideline. The requirement to perform 3 sets of at-rest monitoring was an internal requirement. Through this harmonized approach, could you go back 1 slide, please? Yep, through this harmonized approach, there is a process streamline opportunity where we will be transitioned to performing 1 set for the at-rest, which is supported by the rationale that explained earlier by Gurpreet. And this will help reduce the overall workload, and it is a cost saving. For in-operation monitoring, we perform 3 sets. This is a schematic diagram showing the execution of the EMPQ plan. 3 sets of at-rest is represented on top, and the 3 sets of in-operation is on the bottom. Prior to releasing the room for EMPQ post-construction, we performed 2 acts of disinfection with using phenolic disinfectant and sporicide. Then EM sampling occurred, and then following each run of monitoring, a routine cleaning is performed prior to the next monitoring session. During the execution of EMPQ, there were several protocol discrepancies, and we would like to share 2 notable ones. The first one is 2 differential pressure alarms were activated during run 1 of at-rest monitoring. The root cause was attributed to incorrect instruction regarding door operation were given to the samplers. Thus, the doors were propped open for longer than it's supposed to and activated the alarms. Some action taken include correct instruction was provided to the samplers, Total airborne particulate samples results were evaluated and met the acceptance criteria. Thus, monitoring proceeded forward at the time. Viable air and surface samples were evaluated at a later time and also met the acceptance criteria. Thus, no resampling was required, and this incident did not impact the EMPQ outcome. The next protocol discrepancy, floor surface sample collected from a grade D material airlock exceeded the alert level for all 3 consecutive days of in-operation monitoring. The trend of excursions was due to increased personnel presence and material transfer occurred in the airlock as part of EMPQ setup activities that was beyond routine worst-case operation. The action was to recollect the sample under routine operational condition, and the resampling passed. So the takeaway for this is to consider the impact of EMPQ setup activities on the environmental conditions of clean rooms, as they may not be reflective of the normal operation. And although it was not identifying as a root cause for this discrepancy, an additional consideration is that if site is using contractors to perform EMPQ monitoring, ensure a robust training on appropriate procedures and have appropriate quality oversight. Contractors are initially not familiar with the gowning or material transfer process. Contractors also may use non-site dedicated equipment and more in terms of the number of equipment. So it is very important that we have robust quality oversight. Once all resampling were completed and protocol discrepancy were closed, the clean room were released following completion of review of the data with passing result. However, batch disposition was conditional on completion and approval of the EMPQ final report. In the interim of completion of EMPQ and the approval of the EMPQ report, routine EM was performed, collecting all EMPQ sample sites. Once the EMPQ final report is approved, routine monitoring occurs, collecting a reduced number of sample locations as part of the ongoing monitoring. And now I will hand back to Alice for our closing remark.
Thank you, Luyen, and thank you to all our speakers during this presentation. I hope that it was insightful and great information for you all. Before we wrap up, I would like to thank very, very much all the authors and all the contributors of this work. As I said at the beginning, it is the outcome of many months of discussions and a really active collaborations within BioPhorum. And saying that we continue to work and collaborate on new topics. So I am very much looking forward to sharing more as we go and publish more guidances on EM. So please keep an eye on what's going to be out next. So again, thank you so very much. Until next time, and I wish you a very good rest of your day and a very good rest of your week, everyone. Thank you.